it continues to grow while arrested in meiosis. In meiosis, after one round of DNA replication, there is an induction of double-strand breaks, which some of them are repaired through homologous recombination process. The homologous recombination process requires chromosomal pairing, which is enabled through synaptonemal complex proteins and meiotic coenzymes. The entire chromosomal pairing process requires the chromosomes to move in space in search of their homologue and be brought together. This is why in the lab we move from the textbook view of chromosomal pairing into a more holistic cellular view of the process, in which in liptoten-zygoten stage, the telomeres are tethered to the nuclear envelope via Sankash protein complexes that are in turn connected to the perinuclear microtubules. The telomeres rotate, the chromosomes shuffle until at some point the telomeres cluster to one pole of the nuclear envelope, opposing the centrosome, looping the homologous chromosomes and creating a configuration termed the Zygotene bouquet, because it resembles a bouquet of flowers. The bouquet is conserved from yeast to mammals and is known for hundreds of years, but the mechanical regulation of it has yet to be determined and is unclear. An interesting answer to it came in the shape of our former discovery of the Zygotene Cilium, which is a microtubule-based organelle, 9+0, within the ciliary axoneme here in the ultrastructure. It grows from the centrosome and protrudes extracellularly. The Zygotene cilium is a stage-specific cilium in which in oogonia it's absent, starts to grow in liptoten, in zygoten it's fully elaborated, and later disappears in pachyten stage. And here in live, the presence of the Zygotene cilium in rotating, shuffling chromosomes. The cilium completes the chromosomal bouquet machinery cable system from the telomeres to the perinuclear microtubules emanating from the centrosome that gives rise to the Zygotene cilium. This creates a mechanical cable system resembling that of a Ferris wheel. Our major findings were that the cilium anchors the centrosome, counterbalances telomere rotations, and is required for telomere clustering and chromosomal pairing, hence necessary for meiosis. Not only that, the cilia are entangled in between the oocytes in the cyst, creating a scaffold that is required for cyst morphogenesis and ovary development. So the cilium is an essential player both in meiosis and oogenesis. But what about spermatogenesis? What about mammals and even humans? So our next step was to check in testis. That is organized in seminephros tubule organization, that is the developmental and functional unit of the testis. And we wanted to distinguish the Zygotene cilium from the later forming flagella. To do so, we use SYCP3 marker and Telofish marker, and indeed established that the Zygotene cilium appears and is conserved in prophase stage spermatocytes. But what's the function? And in order to test that, we use two viable ciliary markers, Sec290, KIF7. Both are essential for ciliary formation and function. We 3D measured the Zygotene cilia in the spermatocyte context to establish that in liptoten zygoten it starts to grow, and in zygoten pachyten spermatocytes it's fully elaborated. We wanted to check whether the cilia are completely abolished in the Sep290 mutant background or setup, and indeed there is a gradual loss of cilia upon the ciliary mutants, the Sep290 ciliary mutants combinations. Upon ciliary loss, also the nice tight bouquet configuration of the wild-type spermatocytes is lost in the ciliary mutants. It shows expanded and dispersed bouquet configurations. And consequently, the SYCP3 fails to load in the in the sub-telomeric regions in the ciliary mutants. Hence, it's required and essential for male meiosis. Not only that, instead of a nice, beautiful seminiferous tubule organization in the wild types, the ciliary mutants exhibit abnormal seminiferous tubules and an underdeveloped testis. They have very few mature spermatozoa marked here with the flagella in magenta. And downstream to it, there is no sperm in the mutant's semen. And they fail to fertilize wild-type eggs, both in spontaneous mating and in vitro fertilization. In mice, the cilia are conserved in prophase stage oocytes and spermatocytes, and we wanted to check again the functionality. And to do so, we utilized TALP3 knockout mice that abolish the cilium. The Zygotene bouquet is lost in these knockout mice, and as a consequence, SYCP3 loading also halted and defected in the TALP3 knockout mice. So it's also, the cilium is essential for mice meiosis as well. Both gonads, male and female gonads, testis and ovary, are smaller in the mutants with underdeveloped tissue, gonadal tissue, and exhibit sterility both in male and females. So the cilium is essential for both mice meiosis and fertility, both in males and females. And what about humans? So we checked in 16 weeks post-fertilization human fetal ovaries. We marked for TRF and SYCP3 markers, also with ARL13B, which is a ciliary-specific marker to establish that indeed in human oocytes, in human prophase stage oocytes, the cilia are conserved. And there is a ciliary gene expression signature in prophase stage oocytes in human, both in males and females in RNA-seq assays, which means that the Zygotene cilium could be an interesting essential player for infertility in humans. In that context, we have the ciliopathies, which are a group of syndromes caused by genetic defects in cilia formation and function. Most of the causes are associated with either flagella defects or reproductive tract defects. And the Zygotene cilium is an unknown player, an overlooked player in infertility context in humans. So we wanted to investigate it more. So we checked in patients with premature ovarian insufficiency, meaning that the follicles are depleted or dysfunctional. It has various of causes, but most cases have unknown cause. So we wanted to check for ciliary genes that are in these unknown cause patients. And we've discovered that we have both homozygous and heterozygous ciliary mutations in POI patients with an unknown cause, suggesting that the Zygotene cilium could explain ovarian deficiency manifestations. So what I've shown you today up until now is that the Zygotene cilium completes the bouquet machinery, is considered inherent and essential player in meiosis, and is directly relevant in humans and specifically in women reproductive health. And with that, I would like to thank my lab members. First of all, my PI, Professor Anahi Molla-Herman, and Vinit Kumar, who collaborated with me on this project, Sasha and Kayla, undergrad students who helped enormously to make this thing happen, and our dear collaborators. And my special thanks to the organizers of this conference for allowing me to participate from afar in this very difficult situation. So thank you. And with that, I will take one question.
Well, we have time for 2 questions, maybe. So I can ask a question. Do you hear me?
Yes.
So you are using mutants of ciliary protein, but some ciliary protein have been also involved in assembly of the mitotic spindle, of the spindle. Are you sure you don't have also defects in assembling the division, I mean, for the sperm or the oocyte?
we've established that the basal body, the MTOC part is still functional with the gamma tubulin assay in the ciliary mutant. So it's able to form microtubules and create and be an MTOC, even though the cilia part is defected in these mutants. So we've established it, and yeah, I didn't have time to show it today.
Okay. Is there another? Yeah, there is one question.
Hi. I would actually love to ask when the Zygotene cilium is disassembled in humans, because we know the human oocytes lack centrioles, so it must somehow be disassembled at some point. And when does that happen?
We didn't check. We only checked, like, in terms of stainings and like more elaborated assays. We only checked in healthy humans, not in patients or in a mutation situation.
Because the meiotic spindle in mammals is acentriolar, so the Zygotene cilium should be somehow disassembled at some point.
Yeah, I'm sorry, I'm not sure.
Okay, thanks a lot. Thanks a lot, Avishag. Thanks, and good luck. So Karine Levy unfortunately couldn't give her talk, so we are moving to Stéphanie Rosswag de Souza from the CRG Barcelona.
Okay, thank you. So, thank you. Oh, thank you so much. Thanks. Okay, yes. So, thank you very much. I'm Stephanie, a PhD student at Elvan's lab, and today I will try to convince you that dormant oocytes are sleepwalking. So, we women, we know that we're born with all the egg cells that we'll ever have. We have a cohort of primordial or dormant oocytes that get depleted as we age. And we know that also after adolescence, there is going to be a cohort of oocytes that are going to leave dormancy, activate, grow several fold to give rise to a fertilizable egg. Now, at the lab, I'm working with mouse ovaries to study oocyte dormancy. We know that transcription is active during most of the time of the oocyte growth. We know specifically a lot about this part of the oocyte biology, where the meiotic maturation, because the chromosomes condensate, they stop transcribing. But actually, we don't know quite a lot about this part, specifically where they spend the most time in their lifespan in the dormancy state. So in order to elucidate that, I wonder what's the transcription rate in dormant oocytes. I use 5-ethylene-uridine, which is a uridine analog that can be incorporated into the nascent transcript. And I can use click chemistry to attach a fluorophore to it and therefore visualize transcription rate in the microscopy. So I'm going to show you now a picture of a whole ovary of a P4, so newborn mouse ovary. And I'm using this because they have mostly dormant oocytes. And we see here in blue the DAPI and in green the nascent RNA. And we see actually that the dormant oocytes are transcribing quite a lot, which for me, to be honest, was not expecting that. Then we checked, of course, what is the transcription rate compared, for example, to the somatic cells surrounding the follicle cells, the oocyte, and we checked the ratio. So if the oocytes are transcribing in the similar amount as the somatic cells, we expect a 1-to-1 ratio, and we see that in all stages, newborn, pre-puberty, and young adult, the primordial oocytes are transcribing more than the rest of the cells. We also compare these in the adult ovary. Here we have the primordial oocytes. It's very easy to see because they're shining in green. And we have here growing oocytes, and the nucleus is also here marked. And we compare also the levels of the 5U intensity in the nucleus of primordial oocytes versus the growing oocytes, and we see that actually the transcription goes down as they grow. Now, what are these nascent RNAs? We see that there are 5U oocytes quite visible in the nucleus, so we thought they have to be nucleolar RNA. We checked with the nucleolar marker NPM1, and we see that indeed the 5U co-localizes with the nucleolar marker. So now we have to go back to nucleolus. What is the nucleolus? It's the site of ribosome biogenesis. So here is the nucleolar innermost part, and this is the outermost part. And as the ribosomal RNA is transcribed, it leaves the nucleolus by assembling and making ribosomes. One thing to keep in mind is that ribosomal RNA synthesis is happening concurrently with the ribosome biogenesis, so it's not known that this mechanism happens without the rest. And also ribosome biogenesis is one of the most energy-demanding processes in a cell, so I was wondering, why is a dormant cell doing all of that? First of all, I blocked RNA PO1, which is a ribosomal RNA-specific polymerase, and I see that indeed the 5U foci here, the MSO-treated, compared to the drug-treated one, they are depleted. Here we see the majority of them actually have no 5U foci. And more interesting for me is that the difference that we see before from the oocyte-somatic cell ratio comes mostly from the ribosomal RNA synthesis. Now we go to the next step. We check the ribosomal RNA precursors, and we see that indeed there is the ITS1 here that gets excised as the ribosome matures. It's also more abundant in the primordial sites compared to the growing ones, and also the somatic cells. So indeed, they are also doing this step. And then we also check the last step, ribosome biogenesis, with the ribosomal proteins and ribosome biogenesis assembly factors. We did proteomics in primordial and GV oocytes, and we see very interestingly that here in the heat map, it's easier to see. This is a primordial oocyte that triplicates, and in all of the cases here with the ribosome biogenesis factors is abundant. In the primordial site is either lower or not detected in the GV, M2, or one-cell embryo, showing that ribosomal oocytes could be, showing that the primordial sites could be ribosome factories. Now, the protein the protein translation also takes a lot of energy. They are doing ribosome biogenesis. Are they also translating? So we checked translation in the oocytes by injecting OPP, which is a puromycin analog, and can also be visualized in microscopy with the same method as before. And here, could you please play the video? Sorry. We see a video of a whole ovary of an adult mouse, and in yellow is the OPP, and we see that the follicles are shining, but the oocytes are actually dark, showing that oocytes have lower translation rate. Now, we also wonder if there is any of these features conserved in humans. We have a connection in the Hospital del Mar in Barcelona, so we get ovary cortex of young patients, and here I'm going to show you the 5EU in an ovary cortex. We see that actually in humans, the whole follicle is highly transcribing, with the oocyte also transcribing quite a lot. Here we see the nucleolus also shining up. And if we check the translation rate in humans, here now we see that the follicles are actually shining, but the oocytes are dark, showing that the oocytes are also translating less. So indeed, we have high ribosomal biogenesis and low translation, and this is also conserved in humans. So with that, we see that transcription translation in our case is not coordinated. One thing happens, the other one is not. We have high ribosome biogenesis while we keep lower translation rate in all the oocytes. So I hope I convinced you that dormant oocytes are sleepwalking. Thank you very much for listening to me. This is my lab, thanks to Elvan as well. Yes, Geraldine? Oh, sorry.
Building ribosomes. If you were building ribosomes, you should at least translate ribosomal proteins and low translation. So what you've shown is you're transcribing ribosomal RNA, yes. But what else, is it possible that then it just accumulates in the nucleolus but doesn't actually lead to ribosome synthesis?
So I didn't show this here, but we checked actually we sequenced the ribosomal RNA of primordials and GVs, and there is no difference in degradation between primordials, also somatic cells. So they are not being degraded. It doesn't seem like they're being stored because I checked the 28S and the 18S and I couldn't see granules. They're all very diffuse in the cytoplasm like you would expect. So if they're not being degraded, they're not being stored, they're probably in the cytoplasm. And also I see the 28S and the ITS1 strong in the nucleolus, so they are being made. But also when I check the OPP, I mean here maybe in the image was not clear, but if you zoom in the nucleolus, you see a very low amount of OPP in the nucleolus specifically. So the proteins are nascent and they're going to the nucleolus, also showing that probably they're being assembled. Yeah.
Questions?
Okay, I'm happy to talk later as well. Yes?
Quick question. Have you checked if the oocytes still have nuclear factor 3?
Yes. So I checked so they are in culture and I checked they are nuclear and some of them are not nuclear, of course, because we have all stages, but yeah. So they are dormant.
Perfect. Yeah.
Okay. Thank you very much.
Okay, thanks. So now we have Anahi Molla-Herman from the CIRB at the Collège de France.
Allô, allô, comment ça marche? Okay, good afternoon, everyone. My name is Anahi Molla-Herman, and today I will present to you the work we are doing together with Moji Este, my PhD student in the lab, that I entitled Chromatin Self-Defense Against Transposable Elements, a Role of H2AV in oogenesis and Genome Integrity. Okay, so in the lab I am more particularly interested in understanding how chromatin can regulate oogenesis. And as you all know, fertility can be affected by many aspects of chromatin biology, but today I will only focus in histone variants and small non-coding RNAs. So in the lab, we work with Drosophila as a model system, and Drosophila ovaries are full of ovarioles. And in the most anterior region of the ovariole, you have this fascinating structure called the germarium here. Here, the germline stem cells will divide to self-renew and also to form cysts of 16 cells, and you will have 15 nerve cells and only one oocyte that you can follow. This is not working super well, but I hope you can see that you can distinguish with the help of specific markers like orb here in red. So when the germline exits the germarium, they will get surrounded by somatic cells to form these beautiful egg chambers that will grow until egg formation. And very importantly, the oogenesis can be threatened by a lot of different sources of DNA damage, such as transposable elements. And fortunately, there are a lot of mechanisms that can preserve the germline genome integrity. Very interestingly, the growth and the genome integrity of the oocytes is going to depend on the maternal inheritance of very important factors such as the chromosomal information, the vitellus, and also a fascinating population of small non-coding RNAs such as pyRNAs. Okay, so as you may know, pyRNAs can, together with the PIWI family proteins, they are going to silence transposable elements thanks to the complementarity of sequence and with the help of the presence of the histone mark H3K9 trimethylation and also the help of heterochromatin proteins like HP1. These pyRNAs emerge from the transcription of long pyRNA precursors that also require H3K9 trimethylation and the presence of a very interesting complex called the RDC, rhino deadlock and cutoff. And very interestingly, in Drosophila, pyRNA clusters, they can be expressed either in the soma, in the germline, or in both tissues. And they can also be transcribed as unistrand or dual-strand pyRNA clusters. Recently, it has been shown that the histone linker H1 can also play a function in transposable silencing. And you all know that histones are very important for gene expression. However, little is known about the role of histones and histone variants in transposable element regulation and genome stability. And we have done a genetic screen in the laboratory in order to look for factors that could silence transposable elements, and we found this histone variant H2AV. So in most species, we have two important histone H2A variants called the H2AX and H2AZ. H2AX gets phosphorylated in response to DNA damage, and it's important for DNA repair, whereas H2AZ is more related to transcription activity. And very strikingly, in Drosophila, there's only one H2A variant called the H2AV that is combining both features of X and Z. And very interestingly, in terms of sequence conservation, you can see that H2AV is more similar to H2AZ in terms of sequence, but in the C-terminal tail, there is a phosphorylation motif very similar to H2AX. It's not working anymore, yeah. Okay, so in the lab, we have shown that the mutation of H2AV, specifically in the germline, leads to sterility, oogenesis arrest, and a developmental delay. Thank you. Ça passe ou pas, lui? This is only the laser, or? Yeah. Ah, sorry, I need that. I need it again. I need the other one again. Okay, sorry. So the mutation of H2AV specifically in the germline leads to sterility, oogenesis arrest, and developmental delay. And indeed, with the help of oocyte-specific markers like ORB here in green, you can clearly see that compared to the control situation, where you have a beautiful oocyte determination, in H2AV germline mutant, the pattern is very altered. So what could be the cause? We thought it could be DNA damage. So to address this question, we used RPA-GFP as a readout because it binds single-strand DNA. And in the control situation, we can see that RPA-GFP foci accumulate in endoreplicating cells. However, in the H2AV germline mutants, you can see that the signal of RPA-GFP is stronger all over the ovariole, and there are too many foci everywhere. So what could be the source of DNA damage? To study transposable element expression, we use different complementary approaches, such as RNA-FISH and immunostaining. And here you can clearly see that in H2AV germline mutants, there is an upregulation of transposable elements compared to the control situation. And we also saw these results with RNA-seq. And here you can see that there is a big amount of upregulated transposons in a significant manner, as you see here in red. So if transposable elements are overexpressed, we thought that pRNA should be downregulated. So to study this, we did small RNA sequencing to study first the general pRNA populations. And in these histograms, you can see that there is a mild effect on pRNA general populations in H2AV mutant. However, if you look at this figure here in the right, when we study the specific germline clusters, first the unistrand 20A, you can see that it's not very much altered in the H2AV germline mutant. However, the three most dual pRNA clusters, 42AB, 38C, and ATF, they are severely affected in the H2AV germline mutants. So altogether, we've shown that H2AV mutation leads to a downregulation of pRNAs, the upregulation of transposable elements, leading to DSBs and oogenesis arrest. So which could be the mechanism of this arrest? So to study this, we focused on checkpoint protein activation, since it has been shown that the activation of CHEK2 leads to oogenesis arrest. So remember, I've told you that H2AV mutants have an arrested oogenesis. Now, if you do double mutants of CHEK2 together with H2AV, we have a beautiful rescue of the oogenesis, where we begin to have egg chambers and even eggs. And we wondered what could happen with the downstream factor P53. So to study P53 activation, we used a very elegant tool in which P53, when it's activated, it will drive the expression of GFP. And in here you can clearly see that in H2AV germline mutants, P53 is activated. So altogether, we've shown that in H2V mutant, there is a transposable element overexpression, RNA defects, DNA damage, checkpoint activation of p53 and CHEK2 that lead to oogenesis arrest at early stages. And very interestingly, it has been shown in the literature that the complex of ORB6 together with domino B, they are important to load H2AV into the nucleosome and the chromatin. So to see if ORB6 could phenocopy H2AV, we did mutations of ORB6 in the germline, and we saw that indeed ORB6 mutations have a very similar phenotype to H2AV. And then to finish, since H2AV in Drosophila, we only have one H2AV variant compared to X and Z in other species. To understand the evolution of H2AV, we decided to do a separation of function in which we deleted the C-terminal tail of H2AV. In this way, the phosphorylation motif that is very similar to H2X is no longer there, and the protein is very like H2Z. And in this context, you can clearly see that H2AV lacking the C-terminal tail, they have normal ovaries and they are fertile. And here in the right, you see that there is no longer an accumulation of transposable elements. And these results suggest that the whole H2AV phenotype that I showed you before will rely on the H2AZ transcriptional function. And with this, I would like to thank Jean-René Wien, my PI, and Maud Gineste, my PhD student, and all of you for your attention. Thank you very much.
Question.
Here. Hi. I was wondering, do you see the same transposon de-repression and pyrene production failure phenotype in the somatic cells, or is this specific to germline?
That's a very good question. So the phenotype I showed you, it was only driven in the germline because we use a specific driver. We did similar results in the somatic cells with another driver that is called traffic jam, and we don't see an overexpression of the typical transposons like flamenco cluster transposable elements.
And then if I can follow up with another question. What is the distribution genome-wide of H2AV? Like, do you see any correlation with dual-strand clusters that might explain the specificity?
Yeah, that's a very good question. We are struggling. We are trying right now to do cut-and-run experiment because at this point we really need to know where H2AV is located genome-wide in the chromatin, because we have several explanations. Either H2AV is directly located in the transposons, so you understand that H2AV is no longer there, you overexpress the transposons. Second, the H2AV could be located at the pyrene clusters being required for the transcription. But third, we also think that H2AV, because it has a function in transcription, it could directly transcribe genes from the pyrene pathway, such as aubergine, IGO3, Malmström, rhino, etc. And we have some evidence that it can be a bit of all of them, because in the RNA-seq results, we have genes from the pyrene pathway that are downregulated. So we need to have this data, and it will be very important to answer your question.
Over question? Yeah.
So you mentioned that when you get rid of this H2AV, you could have problems with transcription, like you described, or with like DNA damage response. But then you mostly
Sorry, I didn't understand.
I think you mentioned that because this one gene is taking the function of two genes that have been studied in other systems, right? The H2X and the H2AZ, I guess.
Yeah.
And then you sort of focused on the transcriptional aspect. But are there also problems with DNA damage response from that, for example, when you make that C-terminal mutant that you talked about at the end?
So your question, I don't understand the question.
Are there problems with the DNA damage response? Like what you would see with gamma H2AX?
Are there problems with the DNA damage response?
When you mutate?
So, for example, I didn't test that directly, but I know that, for example, ATM and ATR are the responsible kinases to phosphorylate H2AX, so H2AV in Drosophila. And we could do double mutants, for example, for H2AV and ATM or ATR to see if we can rescue to an extent the phenotype. And another aspect that we have studied for the DNA damage response is that so I don't know if you know the work from Felipe Teixeira. He showed that the oogenesis arrest could rely in a threshold of DNA damage. And we wanted to diminish the level of DNA damage in our H2AV context. So we did double mutants for SPO11, MayW68, together with H2AV. We have less damage, and we are able to partially rescue the oogenesis. So we think there is also a function in DNA damage that we cannot yet understand well, but yeah, it's a good question.
Okay, so maybe we'll go to the next speaker. Thanks, Anahi. So, now it's Noemi Zollo from the CIRB at the Collège de France also.
Okay, perfect. So this works? Perfect. Hi, thank you for the opportunity. I'm Noemi Zollo, and I'm finishing my PhD in the Terre team. Now, my title is quite the spoiler, but pretend to be surprised. In the lab, we're working with mouse oocytes, and it's always important to remind that mammalian oocytes spend a very long phase of growth before they even start meiotic divisions to eventually get ready for fertilization later on. During this extremely long growth phase, oocytes accumulate mRNAs and proteins in the, we've seen already, in the ribonucleoproteins or RNPs. And these, together with the nutrients and all the other cytoplasmic components, make up the maternal inheritance, the maternal stockpiles that will be fundamental to sustain later stages. RNPs in particular are especially important because during growth, transcriptional levels gradually decrease. So meiotic divisions and early embryogenesis entirely rely on the timely translational regulation. Slide. Occasionally, more slide. Aha, yes. Translational regulation of the mRNAs stored in the RNPs. Now, this very long growth phase can last from weeks to decades, depending on the organism, and in mice, it's about 3 weeks. I'm interested in particular in the very last part of this growth phase, which corresponds to the last 2-3 days, and that's because at this point we have a peak in the increase in volume with an accelerated rate compared to the overall growth. And we don't really know how the oocyte achieves this. So, cut to the chase, yes. I identified a novel cluster of mitochondria in late-growing oocytes that was never observed before. You can see here this white spot is the nucleus, and you can appreciate how this compartment is quite big, with an approximate diameter of 15 microns at a stage where the whole oocyte has a diameter of about 60 microns. And it's also transient because the before and after the mitochondria are quite homogeneous. Furthermore, we not only have mitochondria, some remote will have to work at some point. Okay, so it takes some begging. It's not only mitochondria, but also RNPs, as highlighted by the enrichment in mRNAs. So we've seen already with previous presentations how this kind of compartment is not entirely new to oocytes, because pretty much all species have a bulby antibody that is also a membraneless condensate cluster of RNPs and organelles. However, despite the striking similarity in morphology between the bulby antibody from other species and the compartment that I observed in mice, the bulby antibody is found much, much earlier in oogenesis. So what I'm wondering is what could be the role of this later cluster in mouse oocytes. I call this compartment the Zollo body because it has a nice ring to it, so why not? And I started the methodical characterization of its components, starting from a higher level, the organelles, moving down to ribonucleoproteins, and trying to ultimately understand the function. Yeah. So starting from organelles, I already showed you the enrichment in mitochondria, which is my control staining here on the left. And we also have an enrichment in Golgi, ER, with this very peculiar worm-like shape. And we can usually spot an empty region, a hole in the mitochondrial cloud, which is where the mTOC, the microtubule organizing center, is found. The mTOC is the oocyte equivalent of a centrosome, so this suggests a very strong link with the cytoskeleton, in particular with the microtubules. All these findings have been confirmed also by electron microscopy, thanks to the amazing work of Alexis Canet. And we can see how at this stage, pretty much all the membranous organelles are gathered next to the nuclear envelope, that is this wrinkly thing over here. So once again, we have mitochondria that are colored in purple, ER worms in green, and the Golgi mini stacks that are these adorable little rainbows over here that are colored in orange. And once again, we can spot the empty region where the mTOC is found, as highlighted by the enrichment in microtubules. Microtubules that also microtubules that also turned out to play a structural role in the Zollo body, because if I depolymerize microtubules with nocodazole, the Zollo body disappears or anyway significantly decreases its size, as quantified here. What is more interesting, if we get to the slide, yes, is that, no, yes. After depolymerization of microtubules, if I wash out nocodazole, the structure reforms in the same spot next to the nuclear envelope, which indicates that microtubules are not only involved in the maintenance of the compartment, but also in the active assembly. So at this point, we have mitochondria, Golgi, ER, and we discovered an important link with the cytoskeleton, in particular with microtubules, gaining first insights on the assembly mechanism of this compartment. Moving on to ribonuclear proteins, we started from the proteins. Maybe. Yes. We performed mass spectrometry in collaboration with Alvan's lab at the CRG in Barcelona, and to do so, I isolated individual Zollo bodies from single oocytes. This was done slowly and painfully by pipetting individual oocytes in a very thin glass capillary until the Zollo body popped out, sometimes still attached to the nuclear envelope. We can see it here colored for mitochondria and RNAs, while this here that you see in the image is the single oocyte that has just been squished. So it's now emptier of cytoplasm, but probably full of resentment. And most importantly, the fact that this isolation was possible was a big surprise, because it means that the Zollo body is extremely stable. And it is so stable, in fact, that I was able to fix it on a cover slip for immunofluorescence, as we can see here for a Golgi antibody. So from this mass spectrometry, we compared isolated Zollo body with the whole oocyte at the same stage to obtain a list of proteins found in the Zollo body, but also enriched in this compartment compared to the surrounding cytoplasm. If we run gene ontology on this list of proteins found in the compartment, starting from the bottom, we have, as expected, a lot of proteins found in membranous organelles, but most significantly and redundantly, an enrichment in ribosomes, which was quite a surprise. Like, are these ribosome enriched because they are active or because they are stored? We'll see. I validated this list by immunofluorescence, and I moved on to the RNAs contained in the RNPs. So RNA sequencing, as expected, comparing the Zollo body stage with the following stage. We have here the gene set enrichment analysis done by Nicolas, and if we look at the categories of genes that are most significantly deregulated, we have genes involved in mitochondrial respiration, ATP biosynthesis, but the category of genes that is most represented is the genes involved in translation, whether it is enrichment, regulation, or components of ribosomes. So proteins and RNAs, they both strongly point towards the direction of translation. So the next logical step was to check whether the function of this compartment could have to do with active translation. This seems to be the case because we have a positive enrichment, a positive staining for phosphorylated mTOR in the region of the Zollo body, which is the active form of mTOR, providing a link with cell growth and active translation, and it's quantified over here. I also tested I also use the commercial OPP kit, as we saw before. This kit uses a puromycin analog to fluorescently label nascent peptides, so it gives us an idea of local active translation. And once again, we have a positive staining in the Zollo body region. This positive staining disappears if I pre-treat the oocytes with puromycin to prevent translation, so it seems to be quite specific. What's more interesting is that if I depolymerize microtubules to dissolve the Zollo body, the overall levels of phosphorylated mTOR, so the active mTOR, significantly decrease, meaning that the Zollo body seems to have to be intact in order to promote translation. So very cool, we have active translation. What does this imply? If we measure dry mass, which is a non-invasive measurement through transmitted light imaging to estimate the protein content of each cell, we can see how the size of the oocyte spikes after the Zollo body stage, and this we knew. But what the analysis revealed is that this trend is mimicked by the dry mass content, meaning that we have a peak in a significant increase in protein content after the Zollo body stage. What is more important is that this allows for a stable protein density, so we don't observe the cytoplasmic dilution that we would expect from such a significant increase in volume. And since cytoplasmic dilution usually is correlated with the senescence-like phenotype, we can see how this is very important. So, in conclusion, our current hypothesis is that this compartment appears at a critical stage in oocyte growth to boost translation and allow the oocytes to significantly increase their size. So this compartment would allow the protein content to match the changes in volume while preserving a stable protein density. So we're in the presence of a strategical regulation of translation at a specific point in a time point in growth that would avoid cytoplasmic dilution and allow the oocytes to reach the fully grown stage with all the maternal stockpiles that it will need for the following stages, ultimately contributing to the preservation of oocyte quality. If you're interested in the whole story, the article is on bioRxiv, accessible through this QR code, and I'd just like to thank my amazing lab and collaborators. And if you have questions or suggestions, I'll be very happy to take them.
Questions?
Yeah. Hi, thanks a lot.
Thank you.
What happens to the Zollo body at fertilization?
I have no idea. I mean, this is the oocytes with the Zollo body are still 2-3 days away from the fully grown stage. So we don't have the means right now to follow them all the way to fertilization. That's, of course, one of the things that we want to discover.
Hello, thank you for the nice talk. My question is somehow similar to it. So there is also this structure called MARDO in mice, which is found in GBOcytes that also has ER, Golgi, and mitochondria and RMPs. So I was kind of curious if you have any ideas about how this structure is kind of translating into that compartment.
Yeah, so of course it's interesting to draw the comparison because they're not so far apart. So if the Zollo body appears 3 days before the fully grown stage, the MARDOs were described at the fully grown stage. So that's one specific feature of MARDO is the presence of ZAR1 as a nucleating protein associated with the mitochondria, and we don't have an enrichment in ZAR1 at the Zollo body stage. So our findings from the mass spec would suggest that they are not the same, and MARDOs probably don't directly derive from the Zollo body, but I don't know more about the interaction of these two compartments.
Very interesting. I was wondering whether the proteomics also told you something about the mechanism by which microtubules favor this formation.
Yeah, so they didn't tell us much about the microtubules per se. I mean, proteomics told us that there are microtubules, which, yes, we figured. But what's most important is that proteomics allowed us to have a list of possible candidates to look for proteins that might have a similar role to buckyball or X-Velo in Xenopus, so this kind of glue protein that forms an amyloid matrix. Since we can, through mass spec, identify the proteins that are enriched in the Zollo bodies, we can check which one of those have IDRs or prion-like properties to try and understand if those proteins could be candidates and could contribute to the assembly of this compartment in terms of amyloid matrix formation, if that answers.
Related to that, like what holds it all together? I mean, we know about condensates that have proteins and RNAs, as you just said, but then you have organelles in there. Like, how do you envision, I mean, you probably don't know the answer, but how do you envision that, like, it's all stuck together?
Yeah, so I can try to scroll through my supplementaries blindly, but it's easy if I just describe it. So as I was mentioning, the Babiani body in Xenopus and Zebrafish is held together by a sort of physiological glue that forms an amyloid matrix, and this holds together RNAs, RMPs, but also organelles. Since this kind of protein that forms amyloid fibrils can be detected with compounds like theoflavin T, I tested theoflavin T on this stage, of this oocyte at this stage, and I have a positive staining. So there are prion-like proteins that could act as an amyloid, as a physiological glue forming an amyloid matrix. Sorry? Yeah, probably. Yeah, if they are, our idea is that if it's a similar function, now the homologue of X-Velo and buckyball was lost in evolution in mammals, so we don't know if it's exactly the same function, but what we think is that these proteins that have IDRs, they are capable of non-specifically bind to pretty much everything. They're just sticky. And so the idea would be that they might contribute to holding together even organelles.
So your model is that this compartment creates a privileged area for something, translation maybe. So are you suggesting that in the rest of the cytoplasm of the oocyte, there's actually a dilution of something happening? And I mean, have you looked at that? Are ribosomes somehow, as the oocyte grow, the density of ribosomes is lower in the cytoplasm?
So you mean the dilution of surrounding cytoplasm at the Zollo body stage? So meaning that, so I mean, in terms of active translation, based on the stainings that I have so far, there seems to be a difference in terms of level of translational activity in the compartment compared to the outside. Whether the activity outside of the compartment is much different from the following stages, it's not so easy to assess because it would imply a comparison. So from my first quantification, there seems to be a slight decrease in translational activity at the following stage, but it's something that is not so easy to assess clearly.
We have to move on to the next speaker. Maybe you can ask your question to Noemi later. Thanks. So it's Antoine Guichet from the Institut Jacques Monod now.
Okay. Yes. Okay, first I would like to thank Marie-Hélène and Elvan for the organization of this very nice meeting. And so in a cell, and in particular in an oocyte, the tight control of the positioning of the nucleus is a very important event, and it relies at least on two features, which is the process by which the nucleus can migrate and the process by which the nucleus can be maintained anchored. And in order to study, in our team we are interested by the processes by which the microtubule controls those two processes for the nucleus. And in order to study this, pas très bien. Okay, we are using the Drosophila oocyte as model system. So the Drosophila oocyte is a large cell in which the nucleus is asymmetrically located. And during oogenesis, as the oocyte grows, the positioning of the nucleus is evolving. So it's going to be firstly located in the vicinity of the anterior, then it's going to be centrally located, then there will be the effective migration of the nucleus, and then the nucleus is going to be maintained at that position till the end of the oogenesis. And it's important to mention that the positioning of the nucleus is essential for the polarization of the future embryo. So the process by which the nucleus is positioned rely only on the microtubules. And we have recently found that at least two different cues associated with the microtubules control the migration of the nucleus. So one cue corresponds to the centrosomes, and the other cue corresponds to the microtubule associated mud. And those together, they control the migration. But more recently, we have been investigating the process by which the nucleus can be anchored. And this is an important issue because, as you can see, well, I hardly see, but there is a very important cytoplasmic flow inside the oocyte, and the nucleus has to stay anchored in order to resist to this flow. And indeed, if the anchorage of the nucleus is fading, as you can see here, so the nucleus is going to move and to be embarked in the flow. So in order to study by which mechanism the nucleus can be anchored, we first study the connection with the nuclear envelope and the cytoskeleton. So first we investigate the potential involvement of the link complex. It's because I'm not pointing it. Okay. So the link complex, as you have already seen the link complex today. So the link complex is a complex of proteins which span the nuclear envelope and which is tightly associated with the cytoskeleton. And in the Drosophila oocyte, you have one sun protein and two nesprints, MSP300 and classish. And importantly, at the onset of the migration and the anchorage of the nucleus, all those elements are evenly distributed at the level of the nuclear envelope. And it's important to mention that upon the inactivation of the sun protein claroid, both the two nesprints classish and MS300 will not be located anymore at the level of the nuclear envelope. And so upon inactivation of the sun protein claroid, there is an inactivation of the link complex. So by doing so, we could investigate the requirement of the link complex for the positioning of the nucleus. And surprisingly, so that we found that upon the inactivation of the link complex, the nucleus is correctly positioned. However, we did find that there is another important protein located at the level of the nuclear envelope, and this protein is MUD. So MUD is the ortholog of the human protein NUME. So it's asymmetrically located at the nuclear envelope, and more precisely, it's located at the level of the outer level of the nuclear envelope, which faces the cytoplasm. So MUD-NUMA on its own is responsible for the anchorage of the nucleus, but it participates to its migration. And upon inactivation of MUD, the migration of the nucleus is delayed. And interestingly, so when we both inactivate MUD and the link complex, then the nucleus fails to be anchored, and then the nucleus is going to be mislocalized with the flow in the oocyte. So MUD and the link complex somehow interact because upon the link complex, the inactivation of the link complex, the distribution of MUD at the level of the nuclear envelope is compromised. And reciprocally, when you have a mutation for MUD, then the distribution of at least sun is affected. So then next, we wanted to investigate by which process MUD and the link complex in relation with the microtubule controls the anchorage of the nucleus. So we focus on microtubule associated motor, and in particular the dynein motor, since the dynein was new to have the capacity to interact both with MUD-NUMA and with the nesprints in other cellular contexts. And importantly, so at the onset of the migration of the nucleus, the distribution of the dynein motor is asymmetric, like the distribution of MUD. And so we investigate the involvement of MUD and the link complex for the association of dynein to the nuclear envelope. And what we found is that, in fact, we have a collaboration between MUD and the link complex for that. So when we inactivate MUD, so the density of the dynein at the level of the nuclear envelope remain as in Y type. However, the level of the asymmetry of dynein is reduced. When we mutate the link complex by itself, the density of dynein at the nuclear envelope is slightly reduced, but the asymmetry is wild type. However, when we make the double mutant, both of MUD and the link complex, then we have a strong decrease of the level of dynein at the level of the nuclear envelope and a drop of the asymmetry. And importantly, upon inactivation of the dynein motor, there is a defect in anchorage of the nucleus, both by the mean of overexpressing the dynamiting, which is an essential component of the dynating complex, or of big D, which is also an essential component of the dynein-dynactin complex. So the second feature we wanted to investigate for the mechanism by which the nucleus can be anchored, is the connection with the cell cortex. And for this, we focus our attention on the spectraplaking short protein, which is a protein which has the capacity to bind the microtubule and the actin filament, and importantly, has been shown to be in the oocyte to function as a non-centrosomal, to be involved in a non-centrosomal microtubule assembly. And so, interestingly, in the oocyte, the distribution of this protein shot is following the process of repositioning of the nucleus. Prior migration of the nucleus, short protein is located at the posterior of the oocyte. Then when the nucleus is migrating, there is a relocalization of shot, which shifts from a posterior position to an anterior position. And importantly, upon shot inactivation, the migration of the nucleus per se is not dramatically affected. However, the nucleus starts to be mislocalized, and their anchorage is abolished, and this delocalization occurs more specifically at the onset of the cytoplasmic streaming. So, in the oocyte, the positioning and the migration of the nucleus rely also on the signal which is emitted by the follicle cells, which is going to be translated to the oocyte. And this signal is essential for the migration of the nucleus, because if this signaling fails to occur, then the nucleus remains at the posterior. And interestingly, we observe that when this signaling fails to occur, we have an abnormal relocalization of the spectraplaking short proteins, which remain at the posterior of the oocyte. And in this case, we have short protein which accumulate at the posterior of the oocyte at the level of the mispositioned nucleus. So in conclusion, we have identified a dual system to anchor the nucleus in the oocyte. So the first part, at the level of the nuclear envelope, we have an even distribution of the link complex, which is going to function with mud NUMA proteins, which is asymmetrically located, and which is going to restrict in asymmetric fashion the localization of the dynein motor. And at the level of the cell cortex, we have opened the signaling from the posterior follicle cells, a relocalization of the short spectraplaking protein, which is going to be located at the anterior. And then we speculate that this shot is going to function as a non-centrosomal microtubule organizing center, which is going to build a microtubule array, which can be captured by the dynein, which is associated at the level of the nuclear envelope, and this together will lead to the anchorage of the nucleus at the level of the plasma membrane. And so with this, I thank you for your attention. I want to thank first Jean-Antoine Lepezan and Fanny Roland Gosselin and Maxime Pontier, who did most of the work that I present today. And I want to also stress that this work is made in collaboration with the team of Nicolas Minck at the Institut Jacques Monod with Charlotte Mala. And I thank you very much.
Question.
So I was wondering how you restrict mud expression asymmetrically in your nucleus, and if it's really important, so if you can have mutants where mud is localized everywhere in the nucleus, and what would be the phenotype?
It seems that it's not to have, so yes, we have some mutation for this. So it seems what is important is to restrict mud to the nuclear envelope and to avoid that mud can reach, for instance, the centrosomes, because so basically we found that there is two sets of regulation for mud. So we did find the NLSE sequences on mud, which will be required for maintaining mud at the level of the nuclear envelope. But to answer more precisely to your question, we did find mutation in mud which abolished the asymmetric localization of mud at the nuclear envelope, and this has no impact on the capacity of the nucleus to migrate.
Anchor to the cortex?
So we didn't test the anchorage.
Question, Michael. Can you say a little bit about why it's so important to have the nucleus, in the position that it is?
Yeah. Well, I didn't have time to go on this. So basically, in the oocyte, I think for two reasons. The first reason is that during the oogenesis, you have one mRNA, which is essential, which is going to encode for the TGF-alpha, which is going to be located with the nucleus and which be translated locally and which then induce a differentiation pattern at the level of the follicle cells, which is going to be essential later on, early during embryogenesis, to specify the different germ layer and to build the dorsoventral polarity. So basically, if the nucleus failed to be correctly positioned, then the determination of the future germ layer of the embryo will be compromised. And there is a second point which is very important, is that in the oocyte, in order to get fertilization, so the fertilization opened through a small canal, which is at the anterior of the oocyte, and the process of anchorage is going to maintain the nucleus close to this unique sperm entry.
Okay, so if there are no further questions, we move to the next speaker, Yamiini Vadapalli from the Max Planck Institute.
Okay, just checking. Okay. Good evening, everyone. I'm going to talk about my PhD project, where I'm trying to understand how actin microtubules are involved in cooperating this chromosome capture process in oocyte meiosis. Sorry. Okay. So oocytes undergo this specialized cell division called meiosis, where with an aim to produce a fertilizable egg by reducing its genomic content and by preserving its cytoplasmic content. Unlike in mitosis, here the search and capture mechanism by microtubules is not efficient because oocytes are quite large and they have this large voluminous nuclei. So this is where actin comes in play and helping transporting these chromosomes towards the microtubules, and then the microtubules capture them and eliminate the extra genomic content by polar body. The actin function during this chromosome capture process has been highlighted in few species. For example, in our lab, oh my god, what's happening? Sorry. Okay, in our lab, we have already been studied in the starfish how actin is important in oocyte meiosis. Okay. I don't know what is happening. Okay, I should have. Okay, perfect. Okay, so the importance of actin has been highlighted in few species across the evolution. For example, in our lab previously in starfish, it has been nicely showed that in oocyte starfish meiosis, there has been 3 different actin structures which have been found. For example, effectin shell, which forms right before NEBD, and it facilitates NEBD, and then there is this and there is this effectin network. Oh my god, what is happening? Okay, there is this effectin network which contracts and transports all the chromosomes towards the cortex where microtubules are nucleating, and then there are these effectin patches which helps in synchronizing the capture event by the microtubules. Not only in starfish but also in human oocytes. Recently from Melina Schu's department, like in human oocytes and also in porcine oocytes, they have been shown that there are these actin cables which cluster the chromosomes before the spindle assembly right after NEBD and to prevent aneuploidy. And not only in oocytes, recently there is a new paper which actually showed that in early embryonic divisions of mouse, The importance of actin, where the actin helps in centralizing the chromosomes and also stabilizing the spindle to ensure that accurate mitosis. So now, okay, yeah, there has been actin-mediated chromosome capture process, but the question is, how conserved is this actin-mediated chromosome capture process across the evolution? So to study this, I decided to understand this, I decided to study this species Clithia hemisperica, which was nicely introduced by John today morning, which is actually sitting at the base of the evolution. We obtained this model organism from Evelyn Holliston from south of France. So this is a fully grown Clithia which has radially located 4 ovaries, and this is a single ovary with fully grown oocytes and also small oocytes. When you isolate a single oocytes, it's quite transparent, and it is how it looks. And it's quite large, it's around 170 micrometer and a nucleus of 70 micrometer in size. And when we induce meiosis in this, we can nicely see there are a lot of cortical contractions and a lot of cytoplasmic streams that is happening during the whole meiosis. So the meiosis process lasts around 90 to 100 minutes. And to study in detail how exactly the actin and microtubules are involved in this chromosome capture process, I purified a few protein markers. I used Lifact and Neutrophin to stain actin and classical EB3 for microtubules, and we found this nanobody which stains chromosomes. And then when I injected them after purification, we can nicely see the actin cortex, and in oocytes, Clithia oocytes, we observe this nuclear actin localization, which is quite new. And Clithia oocytes don't have a cortically localized centrosomes, but they form de novo when the meiosis is happening. So to look in detail, yeah, when we induce meiosis, there is this actin shell formation, and there are this lot of cortical changes that happening, and then there is this actin network contraction which brings chromosomes closer to the microtubules, and then after they all congress, like after they all captured, they all transported to the cortex within 15-20 minutes. And in case of microtubules, as I said, there are no cortically localized centrosomes, but then they start forming here right after NEBD on the nuclear envelope, and then they move to the center of the nuclear region where they start capturing. This is parallelly happening during the actin contraction, and then once they all capture this, as the chromosome complex has been transported to the cortex for spindle assembly. As I said, there is this actin network. I just wanted to show a nice image because in live cell imaging it's quite difficult to resolve this actin network. So this is a fixed cell showing the phalloiding staining. There you can appreciate the nice actin network inside. And of course we questioned ourselves what is the importance of this actin network. So I did a quick staining with the nuclear envelope, and it's this actin, and I observed this actin spiky structures which are actually poking the nuclear envelope and breaking them. So we believe that the function of actin shell is to also initiate NEBD, as I showed in the starfish movie, that even in starfish we published previously that this actin shell actually forms under the nuclear envelope and then it breaks the nuclear envelope. Now in order to understand what phases are, like where actin is involved and where microtubules are involved, I did 3D chromosome tracking fused with some molecular perturbations. In control oocytes, we can nicely see after NEBD, within 10-12 minutes, all the chromosomes are congressed and then transported to the cortex. When we depolymerize microtubules, we can nicely see that there is no congression happening, like the capturing is happening, but all the chromosomes are transported to the cortex individually. When I stabilize the actin network inside the nucleus using phalloidin, we can nicely see there is no congression happening and there is no transport to the cortex happening. This clearly shows that actin and microtubules are quite important for capturing the chromosomes and also to the transport. To understand in detail what phase is driven by actin and what phase is driven by microtubules, I did some 3D chromosome tracking analysis where you have these 3D tracks which you get from Imaris, and then I try to calculate the average position of all the chromosomes, which is in red, and then you get this average track, and then you calculate the distance of each chromosome traveled to this average track in every time point. Then this gives me a graph like how the chromosome movement, like how the chromosomes are moving over time. And then here to summarize, you can nicely see that in control data, there is this, right after NEBD, there is this gradual decrease in the distance, and then there is this rapid drop in the distance where all the chromosomes are captured and then they are transported to the cortex. In case of no-codazole, we also observe this nice gradual decrease in the distance, but then there is no capture happening, so they are just transported to the cortex individually. Whereas in phalloidin, we don't observe this gradual decrease in the distance, and still there is no, there is no capturing happening, and they are not transported to the cortex, which you can't see here, but yeah. So to conclude that, yeah, this initial phase of capture process is driven by actin, where this actin network contracts and brings all the chromosomes closer to the microtubules, and then the microtubules take over. That's when you see this rapid decrease in distance, like they start capturing the chromosomes, and then once they are captured, the whole thing is transported to the cortex. Okay, yeah, they are transported to the cortex, but how? To understand, like, yeah, as I showed in the Brightfield movie, that there are so many cortical actin changes happening, there are some cytoplasmic flows happening, so we hypothesized, yeah, maybe there is some cytoplasmic flows which are helping transport this aster chromosome complex towards the cortex. Jellyfish has this endogenous GFP, so if you see when you induce meiosis, right after NEBD, you can see that they are just lot of flows below the nuclear region, they are just pushed to the cortex. So I used this GFP as a tracer and performed PIV to understand the flow speed and the flow direction. This is just a preliminary data. I still don't know whether the chromosomes are being transported. I still have to do molecular perturbations to show that yes, this chromosome transport to the cortex is dependent on this, but we can nicely appreciate that the speed and the direction is towards the cortex, which shows that yes, they might be transporting the chromosomes. So with that, I would like to summarize, yes, I've been working with 2 model organisms. Starfish has been very well established in our lab, like there are 3 different actin structures which are forming right after NEBD, and then all of them disappear when all the chromosomes are being transported to the cortex. And in case of jellyfish, we have this pre-existing nuclear actin, which significance we don't know why there is already a pre-existing nuclear actin network. And then these asters are forming de novo along with this actin structures, this actin shell. And then this actin network contracts and brings all the chromosomes to these asters, and then they all transported to the cortex for spindle assembly by cytoplasmic flows, hopefully. With that, I would like to conclude, yes, there are some species-specific adaptations of how actin is involved in capturing these chromosomes, but I believe strongly that this is quite conserved, this actin-mediated chromosome congregation is quite conserved from Cnidarians till humans. Thank you, and open for questions.
Hi, thank you for your talk. I was just wondering, in the no-codazole experiment, when chromosomes manage to reach the cortex, individually or by groups, are they still trying to make a polar body?
This I haven't really checked because I only record till they reach the cortex, but I have to check if they try to eliminate the polar body. I really don't know. I still have to check, but yeah.
Question a bit related actually to the one of Jean-René. So I had the impression that your oocyte was contracting actually before an EBD, but also after an EBD. So where you have this pushing of the cytoplasm, but even before. So did you look at that? And is it what happened also in starfish, where you have this contraction at the time of polar body extrusion?
Sorry, again.
In starfish also you have this contraction that goes along the cortex. So is it the same in Clitia?
During the polar body elimination or?
No, just before an EBD.
Before, I think there is also a little bit of, I believe that there is also some cortical contractions during this actin network contractions or right after NEBD. I observe these flows, but I haven't yet characterized. I'm right now focusing just on the flow of transport to the cortex, but then I also observe this, there is this backward flow of the, which is pushing or creating some flow during this NEBD. Yes, there are some flows, but I haven't characterized yet.
Thank you very much for your talk. I had a question which is also a bit similar for the last 2. It might be just a very naive curiosity question. Because you talked about contraction and this actin meshwork contracting and pulling the chromosomes. Did you look at myosin 2 by any chance, or myosin to see if that motor was responsible as well?
Did I look what?
At myosin.
Myosin, no,
Yeah.
I haven't looked yet. Probably that is the plan. I will be doing those experiments as well, yeah.
Okay.
Yeah.
Thank you.
Actually, I had a question. So your lab previously shown beautifully that the centrosome in starfish get progressively eliminated during. So is it the same in Clitia?
No, as I said, we don't have active centrosomes, but then somehow the centrosomes start, like the asters start forming on the nuclear envelope. When I also try to stain for gamma tubulin. We always see dual asters all the time, and there is also gamma tubulin staining. So I believe that they are getting activated, but again, at the spindle assembly, they are just disappeared. So it's.
So you've done, there has been EM done on this oocyte to show that there are no centrioles already.
I think there are centrioles. They are getting activated and they are disappearing immediately after the capture process. So, yeah. Because the spindle assembly is acentrosomal. They don't have any centrosomes in the spindle assembly.
Okay, so it's different from starfish then?
Yeah.
Okay.
Yes. Yeah.
Or a question?
I just had one question. Did you look at the microtubule network in case of stabilization of actin or the reverse to see how it affects the other network that is remaining in your condition where you use drugs, you know?
Again, sorry?
So when you use nocodazole, did you look at actin, how it was looking like, and the reverse, like when you use stabilizing, I guess, with lateral?
I haven't done that yet. I have to do those experiments, yeah.
Okay, so if there are no further questions, let's thank all the short talk speakers who have attended the talks. And now we will have the last speaker, last but not least, Andrea Pauli from the IMP in Austria. And we hope that this thing is going to work.
This up. The two. Yes. Well, thank you very much for everyone for staying. So, and thank you to Marie-Hélène and to Elvan for inviting. So I would like to talk today about one topic in my lab that has been really fascinated us, and that is how fertilization works. I think so far in this meeting we have heard a lot about dormancy, about RNPs, about granules, about how the nucleus positions itself. We are also working on some of these topics in my lab in the dormancy, but I would really like to spend the next 23 minutes on speaking about fertilization, because fertilization is a process that you might all think that we know a lot about it because it's so conserved, and all organisms that are sexually producing absolutely require this step to actually work. But it's very surprising that we know very little about the molecular mechanisms that trigger this step. And we are studying this in zebrafish. So zebrafish is amazing for this. As you heard already, you can have external fertilization, so meaning sperm and egg need to find each other in the water and meet. And we can also take movies of this, which Victoria, she's actually here in the audience, also has made this beautiful movie. And I think it really captures what's happening here. So in yellow, you see the sperm. In egg, this is the whole egg, which is huge. You have calcium waves happening during this time, and then you have this outer shell, the chorion, which breaks away, and the single site where the sperm had to find it and enter, that is where basically the process of fertilization happens. And while many of you might now think, oh, all these dynamics is of course fascinating, I would like to focus you for the next 20 minutes on this process here, what happens at this site when sperm and egg meet, and what do we know about this. So we stumbled into this process very serendipitously with my first PhD student in the lab, Sarah, who generated a mutant for a gene that we've discovered with ribosome profiling that was translated in the oocyte. And this gene was not known before. We called it bouncer. It was a short open reading frame, only 80 amino acids is the mature sequence. And what turned out to be extremely interesting is when Sarah generated a mutant for bouncer in the female, she realized that all these eggs that were laid by the female are completely unfertilized. So these females are completely sterile, the male are completely fine, and this RNA and protein is actually specifically made in the egg. I'm not showing the data for this because this is already a long time ago. We also found out that bouncer is actually required for the sperm to bind to the egg. So it's really on the surface of the egg, and the sperm can bind in the wild type situation, but not anymore in the mutant. So it was the first factor that we discovered here that is essential for fertilization in zebrafish. What was interesting, what immediately caught our attention, that while bouncer is present in all vertebrates, or a homologue of bouncer, in fish particularly, there is actually quite a lot of sequence divergence between different bouncer proteins, between different fish species. So this is actually the mature full-length sequence that I show here, that is the bouncer protein. And that made us think about a crazy kind of idea whether bouncer might not only be an essential for sperm to enter, but might also be playing a role in species specificity of fertilization, given that fertilization happens outside of the body of the female, so meaning sperm and egg need to find each other in the water. And to test this, we turned to Medaka and zebrafish as a pair, because these are two evolutionary really distant fish species. They're 150 million years apart. The bouncer protein is only 40% identical between them. And most importantly, when you mix zebrafish and Medaka gametes like sperm and egg, they cannot normally cross-fertilize anymore, even in vitro. So meaning there's no recognition between them. And what we hypothesized is really this crazy idea that maybe by switching the bouncer protein, like normally the bouncer for Medaka is expressed, of course, on the Medaka oocyte, if we were to express it on the zebrafish oocyte, would we now allow Medaka sperm to enter the zebrafish egg? And this experiment was done by another PhD student in my lab, Krista, at that point a master student. And what I'm going to walk you through this experiment because I think it's really revealing. So in a normal situation, you have zebrafish bouncer on the zebrafish egg, and zebrafish sperm can fertilize, but Medaka sperm cannot. So this was not surprising. This was known. When we don't have bouncer protein on the zebrafish egg, none of the sperm can get in. Yes, it's essential for fertilization. And what was a really surprising result was when we now express Medaka bouncer on the zebrafish egg, Medaka sperm can fertilize, but zebrafish sperm can no longer get in. So this experiment really allowed now the wrong species sperm to fertilize the zebrafish egg. These eggs can even develop, so we now have a hybrid embryo that has half of the genome zebrafish, half of the genome Medaka. They don't get very far, but they get to roughly 24 hours, and they even form an axis. So you see here a head structure, a tail structure, and then they arrest. We do know that the switch on the transcriptome, both the Medaka genome and the zebrafish genome is switched on with the timing of the zebrafish egg. So that's another question that we have. It's a very interesting model now to study also parental effects in the embryo, because we can also make the reverse hybrids with exactly the same trick where we make this Medaka eggs and have the zebrafish as a sperm. But for the purpose of what we learned now about fertilization, what this experiment told us that bouncer ensures that sperm and egg are compatible with each other during fertilization. So meaning here, just to recap here, the zebrafish sperm can only fertilize an egg that has zebrafish bouncer, while the Medaka sperm can only get in when the Medaka bouncer is here and there's no cross-fertilization happening. So this experiment, if you think about it, already told us something else. So they told us that bouncer is extremely important in fish for fertilization, but it also told us that there must be something sitting here on the sperm that is recognized by bouncer. Otherwise, this experiment wouldn't make sense. So there must be something that is different between zebrafish and Medaka sperm that bouncer recognizes in a species-specific manner. And in the past 4 to 6 years, we have roughly spent to try to figure out what could be this question mark here on the side of the sperm. And this was a more difficult project than we had initially thought. So I would like to share with you what we found. So in fish, I mentioned already, we only had the single factor, bouncer. So this was our starting point. So what could be sitting here on the side of the sperm? And we turned to mammals to look at mammals to learn something from the mammalian side, because in mammals, by now, already quite a lot of factors had been shown to be essential for fertilization, meaning taking out each one of these individually in knockouts in mice results in complete loss of fertility, either from the side of the male for these factors or from the side of the female for these factors here. And interestingly enough, some of these factors are conserved in fish. So they are conserved actually in all vertebrates, and these are particularly SPACA6, Isumo, and DCSD1 and 2. And I will get back to these names later, so it's worth remembering them. So these factors are not only present in fish, but we could show by knocking them out, they are also essential in fish for the process of fertilization. So the simple model would be, okay, maybe one of these is actually the direct ligand kind of for bouncer here from the side of the egg, and that would mediate, for example, adhesion. That turned out to be not the case, so we tried to express Isumo from Medaka, Isumo from SPACA6 from Medaka on the zebrafish egg. This didn't have the same effect that switching bouncer has. So it wasn't as simple that it's a simple kind of just swapping the receptors here. So what do we make out of this? So the main question that we had, given the situation in mammals and the situation in fish, is even for the factors in mammals, we lack a molecular and mechanistic understanding what each of these factors is doing, except for this one pair, Isumo and Juno. This is known to be an receptor-ligand pair, and that mediates the binding between sperm and egg in mammals. But all the others, we really don't know what their function is during the process of fertilization. So we were thinking, and there was some indications already, that maybe these factors would collectively mediate fertilization, for example, by forming a bigger protein complex. We, of course, also had in mind that maybe we are missing some other factors that haven't been identified before in mammals, and they might be either fish-specific or they might actually be also conserved in mammals. So this project was led by two fantastic people in my lab, Victoria and Andreas, and I would like to share with you what we found. So and the key to this, the starting point of this, basically how we figured out what is on the side of the sperm, was an AlphaFold screen. So I think AlphaFold for us was a complete game changer because it allowed us in silico to predict whether any of these known sperm factors would interact, at least predicted to interact structurally, and with any testis-expressed membrane and secreted protein. And I should also give a shout out to Clara here in the audience, who actually helped us during the time as a student in our lab, as a master student, to actually run these very first AlphaFold screens. So in this AlphaFold screen, I would only like to focus now on Isumo and SPACA6. So what we did, we asked whether pairwise, basically Isumo or SPACA6 would interact with any of these other 1,400 testis-expressed membrane proteins. And the result was very revealing. So Isumo, so what is shown here is an IPTM plot that gives you the confidence in how much AlphaFold believes that an interaction would take place. So meaning high here means that there is a high confidence that Isumo is interacting with SPACA6, and similarly SPACA6 was predicted to interact with high confidence with Isumo. This was really interesting because experimentally people have tried to show that Isumo and SPACA6, these ectodomains, would interact and could never verify this. So experimentally they hadn't found that there was high enough affinity. However, AlphaFold was completely sure that these two proteins should interact. And what was also interesting, and maybe even more interesting to us, was there was a new factor that popped up, TMM81. No one had ever looked at this. That was also predicted to interact with high confidence with Isumo. And when we now put all three together, we actually found that these three factors were predicted to form a trimeric complex. And this is not only true in fish. We ran the screen in fish, but we also then, of course, predicted it for all other vertebrates. And this is also true for human, for mouse, for all other vertebrates. So this was extremely exciting because now we had here a trimeric complex that could maybe give a function to these proteins where we didn't know what they are doing. And we had a new factor, TMM81, that no one had ever looked at. So the immediate questions that came up from this is, of course, like whether TMM81 is indeed a new essential factor for fertilization, and secondly, whether this trimeric complex indeed exists and what its function might be. So the first is very simple. We generated a knockout in fish and realized that males are sterile, females are completely fine, exactly how we had predicted from a sperm-expressed transmembrane protein. Our collaborators in Japan, in Masahito Okawa's lab, in particular Yonggang Lu, did amazing work there in mouse and also generated mutants in mice and could show that they're completely sterile as well. So meaning this is a conserved factor that is not only there in fish, but actually also in mammals required on the side of sperm for fertility. So we could, there should be here tick boxes, so this is ticked. So this is basically, it's a conserved factor. So what about this trimeric complex? We had already an indication from the fact that all mutants that we had looked at that are forming this trimeric complex, they share exactly the same phenotype. So they are arresting at the same step during fertilization. However, more clearly, kind of in terms of pointing towards a complex formation, was this finding that we realized that when we remove one factor in zebrafish genetically, we generated a mutant, we lose the other two factors also from the sperm. And that experiment I can share with you here. So we used mass spectrometry, a very sensitive way of detecting these individual peptides because they are low abundant on the sperm. So, but in a wild-type situation, using this very specific technique, we were able to detect all three proteins, TMEM81, Isumo, and SPACA6 in fish sperm. And that is what I already mentioned, like if in each one of these mutants, no matter whether we have TMEM81, Isumo, or SPACA6 knocked out, we lose all three proteins. So that is a clear indication that they might actually need each other to stabilize each other. It doesn't necessarily mean that they form a complex. However, we had one experiment, and that was Victoria with another master student in the lab actually performed this, where they tagged TMEM81 in zebrafish sperm, lysed the sperm, and then pulled on TMEM81 to ask which other proteins would be co-IP'd. And we were absolutely stunned when we found that the top three enriched proteins after this IP are our trimeric protein complex members. We wouldn't have believed it, I would say, given that these are membrane proteins, but this would work so well. But here, these were really the ones that were enriched after this IP. We also have evidence from human cells when we express these human proteins, here these sperm factors in human cells, we can also ectopically express them and find when we IP that we again enrich these three proteins in an IP. So with that, we could actually update now the model, how we think about this fertilization synapse. We usually call it synapse. I didn't introduce it like similar to a neuronal synapse, where these two membranes need to come very close together, that we can add TMEM81 as a new factor in both mammals and in fish. And not only we can add it, but we actually have evidence that they come together to form a complex. So now I come back to the questions that I started with. Who is now this interaction partner of bouncer on the sperm? I haven't actually solved this riddle to you. And we were, of course, not only running our AlphaFold screen for the sperm factors, but we also included the egg factors that we had here. And to our big surprise, bouncer was predicted with really high confidence to interact not with a single one, but actually with this interface formed by SPACA6 and Isumo. And that's why our individual experiments did never work. So it is predicted to bind in the cleft between Isumo and SPACA6 that is only formed by forming this trimeric complex. So here's a higher resolution prediction. And that, of course, is only an AlphaFold prediction. So do we have any evidence that this is really the case? It fit extremely well to all the experiments that we had done and the experimental data, but we wanted to have experimental evidence. So we expressed the zebrafish sperm factors in HeLa cells and then incubated these HeLa cells with a tagged version of a recombinant bouncer protein and then asked under non-polarizing conditions to which cells did bouncer protein actually bind. And in this situation, like if you don't express any sperm factors, bouncer is not labeling these cells, but if you have these three proteins, the zebrafish sperm factors present now ectopically expressed in cells, you get this nice staining of the surface of the cells with bouncer protein. And this depends really on the presence of these two factors that form the cleft, Isumo and SPACA6. In cells, funnily enough, you don't need TMEM81. So TMEM81 is really only necessary in vivo, we think, or at least mainly necessary there to stabilize this complex formation. We could then also generate mutants specifically to disrupt this, to test more whether the structural prediction is true, and this holds actually true. So overall, what we think is that fertilization is an amazing playground for evolutionary innovation. So if someone had asked me before, I would have said that the sperm is probably the evolutionary divergent one that went through a lot of changes and has rapidly changed. What turns out to be the case is that the factors on the side of the sperm are actually conserved between mammals and fish, while the egg factors, in this case here Juno, that is the binding partner to Isumo, and bouncer share really no homology, and they are completely unrelated evolutionary. So this is very interesting that here this conserved sperm factor uses different egg adapters in order to achieve the same thing, namely to bind to the egg. In mammals, there was another paper that also predicted, similarly to us, did AlphaFold predictions, and they predicted for the mammalian complex also that CD9, which is a tetrasparine protein known to be involved in fertilization, would bind exactly at the site where bouncer might bind. Whether this is really the case, future will tell. We don't know yet, but I think it is extremely exciting that we have here now molecules and can actually put functions to them. So there are many open questions and loose ends. So I told you where we are right now. But of course, the story doesn't end here, and you might have many questions in your own head by now, and I'm happy to discuss further about this. So one is, of course, this equivalence between Juno and bouncer. So evolutionary, I mentioned there's nothing similar, structurally completely different, but we predict that evolutionary or functionally they might have the similar role. There might just be an attachment point for the sperm trimer to bring these two membranes to adhere to each other. So that's a prediction that we have. The other one is, of course, that this sperm trimer should, in principle, also be species-specific between zebrafish and Medaka. We have tried to test this, and it's extremely difficult to test this because we probably need to change all three proteins. So changing a single one is not actually doing the job because the Medaka proteins don't interact well with the zebrafish protein, so it's way more complicated than just on the side of the egg to change it. And the holy grail in the field, I think, is really the question of fusion. So I told you a lot now about how sperm and egg might bind to each other, and we are thinking a lot about how could this protein complex now between these two membranes lead to fusion. We don't know, that's the short answer. That's how much I can tell you, and I can tell you a little bit more. And so what is really thinking about is interesting. This alone, like when you have this protein complex there, this would almost prevent fusion, what we say, because actually you need to bring the membranes way closer together in order for fusion to arise. So how can we think about this? So there are a few other proteins that we have to play with that I basically didn't mention so far, very much in my talk. In mammals, you have all these proteins. In fish, you have these ones that are all conserved. We already made knockouts for these. And what really stands out to us, and maybe also to you from this picture here, is DCST1 and 2. These are conserved not only in vertebrates, but actually down to C. elegans and Drosophila as well. And they don't really resemble any fusogens, like viral fusogens or proteins, basically, that are known to be sufficient to cause fusion, but they have certain features that are making them particularly interesting. And why do I say this? So in cell fusion, we know from quite a lot of already from the viral fusogens, of course. These are these spike proteins, the structures are solved, placental formation, also uses viral-like fusogens, the syncytines, some people of you might know about these. Muscle development uses fusogens from a completely different type. Fertilization in vertebrates, we don't know what the fusogen is, so this is the big question in the field, and there hasn't been a single protein identified. However, in plants and in algae and protists, there is a single gene, HAP2, that has been identified to be sufficient and necessary for the step of fusion. And this is very similar to these viral fusogens. So when we now look evolutionary on a tree, so this is a phylogenetic tree of the known fertilization factors that we so far know, you might be able to recognize that HAP2 is here in these plant protist lineages. Also here, some species have it. But DCST1 and 2 really stand out as being the most conserved from all the other. These are the other vertebrate-specific factors, versus DCST1 and 2 stand out. So we are at the moment putting our money somewhat on this machinery by this ancient complex, which no one really understands how it works. It has a hydrophobic tip here at the front, which makes us even more excited about working on it. And so what we are at the moment, we are more like fusion dreamers. As I say, we don't yet know how it works. What we think might be happening that this trimeric complex that we've been studying so far might be the recognition adhesion module, while this one might maybe trigger the fusion process. And that is where we basically have a lot of hand waving and a lot of question marks. However, we have one very exciting piece of information, and we always thought like of these entities as separate because we have AlphaFold does not predict interactions between this complex and the DCST1 and 2. The mutants, however, share very similar phenotypes again. And we are therefore extremely excited. So this is the last slide that I have. We have identified actually 2 new factors that bring this whole complex together to form a larger heptameric complex. And that is what we are currently working on. So we think that that might tell us more about the fusion. We don't yet know. So this is where we are at the moment. But it gets more interesting, more complex. And definitely there are more factors playing a role. And we think that they might all play together in order to trigger fertilization. And so with this, that is where we are. So we would love to know here what the key on the sperm is, also what the receptor kind of, how does it work on the egg side. And that's a shout out to Marcia, who made, I think she is here, who made this amazing artwork for us. Like, we would love to shed light on the molecules that are here on the side of the sperm, but also on the side of the egg that allow then the door fertilization to open, that the sperm can actually get in and these 2 very different cells can fuse. With that, a big shout out to my lab. It's a fantastic team. We have a lot of, like, this is master student, bachelor students at the moment in the lab. And a big thank you really to everyone who has helped, who has contributed over the years. Our collaborators, particularly Masahito Ikawa in Japan, who has been absolutely instrumental, is also now still absolutely instrumental in the future. And then all the other people that helped along, and also funding sources, and I'm happy to take any questions. Oh, this is a bad one, so I go back.
Question.
So you were saying that the bouncer sequence is very divergent between Zebrafish and Medaka. So it is the same for these isomos, PACA and TMM, and we have coevolution. Can you imagine that you have coevolution in this species?
Yeah, so they are not as divergent. So they are overall much more conserved, because also bouncer is way more similar between different fish species, but still different. So we did look at isomos, PACA6, and TMM81 as well. And what is interesting, what I didn't mention, is in the experiment that we tried, SPACA6, when that we can replace the Zebrafish SPACA6 mutant can be fully rescued with the Medaka version of SPACA6. So this is completely compatible with forming this complex. However, Medaka Isumo is not compatible. So Medaka Isumo completely fails to rescue. So we think the species specificity must be in the Isumo region. But yeah, it's a bit more complicated than this, but very simply speaking, so it is more likely here that this is here in the tip of the Isumo region. Really nice. I was wondering, so bouncer, when you found it, you said it's also, I mean, you have, it's not well conserved, but it's present also in vertebrates, right? Yes. In mammalians. Yes. But there we have Juno, so how does that fit? Do you say, I mean, what would it do there? Do you have any idea? Or? Yeah, we have an idea. So we were very puzzled by it, because in mammals on this sequence alignment that I showed you, there was reptiles, there was mammals, and what is extremely interesting that the mammalian homologue of bouncer is called SPACA4. That was an annotated gene, and SPACA4 stands for sperm acrosome associated 4. So this never made sense to us. So in mammals and in reptiles, it is expressed exclusively in the testis, while in xenopus and in fish, the bouncer homologue is expressed exclusively in the egg. So we generated knockouts in mice to test whether it would also play a role in fertilization in mammals, the SPACA4 homologue, and indeed, they are subfertile. So there it's required from the side of the male and not in the egg. What is also interesting, it is required from the side of the male, and it's required for a different step of fertilization. So in fish, it is required for the sperm-egg membrane interaction. In mammals, when it comes from the sperm, they fail at the step of zona pellucida binding and zona pellucida penetration already. Once you remove the zona pellucida in mouse artificially, like then the sperm, the knockout sperm can completely normally fertilize. So it's a different step of fertilization, but it's the same, it's the paralogous protein. And it has nothing to do with the trimer binding. AlphaFold also, when we try to predict SPACA4 to bind to the mammalian trimer, it's now both on the same side. It doesn't predict it to interact. All right, so fertilization is more than just 2 membranes fusing, right? There's a lot of signaling, which is where this idea of the fertilization synapse being similar to an immune synapse or a neuronal synapse, right? So could it be that the Izumospacassix TM81 is more involved in the signaling bit, which, because during fertilization you need to signal to the rest of the oocyte to resume meiosis, to make all kinds of secreted molecules to prevent other fertilization events, etc.
Yes, right? So we would love to have a better handle on this question, I would say. So we do know that these have very short cytoplasmic tails, our 3 sperm factors here. And in the case of Izumo, they have actually, in mice, they have removed the cytoplasmic tail. That's, of course, on the side of the sperm, and there was no defect. So from the side of the egg, so far, like the bouncer protein is a GPI-anchored protein, so it has nothing that goes into the cytoplasm. In mammals, there is CD9, and that is a tetraspanin protein, so that could do some signaling to the side of the egg. However, so far, we don't know. So there might be some proteins that signal to the egg, or it could really be that once the sperm, these membranes fuse, that would cause the signaling. But we don't know. So from the side of the egg, we really have not much of a handle, because Juno is also GPI anchored. So both of the adapter proteins at the moment, they are both GPI anchored, so meaning they don't have a cytoplasmic tail that could signal directly to the egg. However, these sperm factors, after fusion, they could, of course, they might end up in, or probably they do end up in this egg or in the zygote cytoplasm then. And these here are ring domain proteins that have a cytoplasmic ring domain, so that could potentially then do something to the signaling in the egg cytoplasm or zygote cytoplasm, but we don't know. So it is known from C. elegans, actually, I should say, that these are required for fertilization. So from C. elegans, they made mutants here. That is known that the ring domain is essential for the process of fertilization as well. So that's the only case for all of these fertilization proteins where cytoplasmic mutation has actually generated a defect. But that's on the sperm, the cytoplasm of the sperm. Peter?
Did you have a look at some rapidly diverging species? If you
Oh, sorry, I did not see your floor. There was the microphone was not there.
Now I'm confused. Did they just take the microphone from you?
We have flow afterwards.
Okay, sorry about it. Did you have a chance to look at some rapidly diverging species to see, with AlphaFold at least, incompatibilities and where they would appear? Like the African lake cichlid or something like that?
So we did look in cichlids. The bouncer proteins in cichlids is almost identical. So they are clearly, there is not enough divergence between them. In other cases, we have not looked, but I think in most cases, in particular cichlids is maybe a good example. I think sperm and egg, if you do mix them in vitro, like they would still work. So I think there's lots of other steps behaviorally that would make species go apart. And we also don't think that bouncer is a speciation factor. I should point this out because we have also evidence the zebrafish sperm, for example, still works with the seahorse version of bouncer. So it is not, we were extremely lucky to pick this zebrafish medaka, but in many cases, if it's still compatible, it works. So it's not a speciation factor. Its main function is really to allow the sperm to get in. So maybe that answers the question. But it is, of course, creating, like the compatibility needs to be there. And if it's not matched, then it doesn't work. In mammals, Juno is also known to be species specific.
So Andy, I have the question about, you showed us the very beautiful micropyle of zebrafish, and other fish have micropiles too. And so how much are interactions, with the micropyle versus with the egg cortex? So if you take off the chorion, do the sperm have an independent way to fertilize? Or is it solely, are there different interactions that you might have to account for the shape of the micropyle in the sperm?
Yes. So in our case, in the bouncer mutant, the micropyle is still completely normal. And we do know when we remove this chorion, this outer shell where the micropyle is formed, in a wild-type situation, the sperm can still fertilize, but then it can basically fertilize at any point. So it really, the micropyle is there to only allow one sperm to get in. In the bouncer mutant, there's no fertilization happening anymore when we remove the chorion. So these factors that we have on the side of the sperm, we have at the moment, so we don't think that they are necessary to find the micropyle or to even get there in first place, because we have no defect in finding. We call it like the approaching the micropyle, and none of these mutants, this is defective. So there might be some attractants released, and there is data out there that there is some proteinaceous matrix kind of on the micropyle that attracts the sperm there, that we have not identified what molecules these are. We don't know. We don't think any of the proteins that I talked about today have a role in this process, because all of our sperm can still perfectly find the micropyle, it just doesn't get in anymore.
Medaka sperm get in without the...
Yes, the Medaka sperm also perfectly finds the micropyle.
Without the chorion.
Yeah. Without the chorion, I'm not sure whether we tested it.
Okay.
That I think we might not have done. I don't think we did that experiment. I think we tested with a micropyle and it finds it and it can get in.
I actually also, there is no question. I had a question. So when you did this experiment with the Medaka bouncer into the zebrafish, you actually didn't do it in a bouncer knockout context, did you?
We did. In the bouncer knockout, there's none of the sperm gets in.
Okay.
So in the bouncer knockout, you do need a bouncer protein there in order for the one sperm to get in.
Okay.
Thank you. So I was just curious, like, is bouncer everywhere in the... Like, or is it kind of localized to a specific region?
Yeah,
no, because like in a conversation about the micropyle.
Yeah, so we have a crappy bouncer antibody that we made ourselves that's not good enough for IF. It works on a Western, but for IF it doesn't work very well. We made a transgenic line that has a chief PTECT bouncer, and that is fully functional. And that, we looked at the localization of the bouncer protein, and it is enriched at the micropyle, so there's more, but it also is surrounding it. So, and that would explain why in a wild-type situation, when we remove the chorion and this micropyle, sperm can actually bind to the entire surface of the oocyte. So we don't really think that the bouncer is only localized at the micropyle. There might be more there. We would love to know how many molecules. We don't. We have no clue how many of these proteins you actually need for this process. We don't. But there is more spread out, but based on our overexpression actin promoter line, there's more at the micropyle.
Okay. So I think we should thank all the speakers for today. Thanks, Andrea. So this closes the day. So tomorrow we start at 10:30. And so the first speakers could come a little bit earlier to download their presentation. And so for tonight, we have organized a dinner only with the invited speakers. It's at Restaurant Le Coq. I think I sent you the address, and it starts at 7:30 p.m. Okay? So see you tomorrow.